Definitely not my favorite. Any ideas why it would not be my favorite? There's no structures, yeah. So, but again, that's kind of typically how this section is taught. You aren't responsible for the structures, you're responsible for knowing all the reagents and all the steps and what enzymes and all that fun stuff. So yes, you're responsible for that, but I'll give you structures at the same time. Okay? Because what I want you to be able to do is to prove that you understand what's going on, or give you the opportunity to prove that you understand it, and that's where I'll give you structures. If you don't want to understand it and you want to rote memorize, that's fine too, because I'll give you the structures and the names of compounds, and if you can recognize and have memorized it, awesome, good for you. You're better than me. Okay? So, that's got it all kind of written out there. Anybody memorized it yet? Okay, good. Because then I can ask this question. The only thing we got are the enzyme names and the step in the Krebs cycle, or citric acid cycle. So, what could we possibly ask? What is the important thing to get out of the citric acid cycle? Not ATP. ATP actually does not come into or out of the electron carriers. It's our NADH and our FADH2. Okay, so what question could I ask?
How do we get there?
Which of those reactions involves NADH or FADH2, so an electron carrier? That's a nasty question. One that I probably wouldn't ask because I think that's a bit over the top.
How do you figure that out from the enzyme? Or are you telling us to memorize what happens?
Remember, we've talked about an enzyme name. So if we saw kinase, what do you know?
Doesn't it deal with redox?
Kinase was the transfer of a phosphate using ATP. So if you see a kinase, you know ATP is involved, and it's transferring a phosphate from one location to another. We've talked about some other enzyme names. Dehydrogenase. Or at least that's how I say it. I don't know if it's correct. What is happening in a dehydrogenase? Getting rid of hydrogen. And the element of hydrogen is H2. If I get rid of hydrogen, what am I getting rid of? I could say I'm getting rid of a positive hydrogen. Could I get rid of a second positive hydrogen to make hydrogen? Sorry, let's rephrase that. Could I get rid of a second hydrogen atom that is positive to make elemental hydrogen? Why not? If that was positive, well, that's not elemental hydrogen. For it to become elemental hydrogen, what do I have to get rid of? A negative. I need a hydride. Is hydride stable? No. What do I need? A redox mediator. So anytime we see dehydrogenase, we're looking at probably so at this point we got to be careful, because remember, the directionality associated with the name isn't necessarily true. Okay? So when we see dehydrogenase, we need to say we need a redox mediator. Okay? Because if we add on top of that these are being used in the citric acid cycle, what we're producing with these dehydrogenases is NADH. So they will consume NAD+ and make NADH. Okay? So dehydrogenase just means redox stuff is happening. Usually our redox mediators, NADH or FADH2. Okay? If we layer that with the citric acid cycle and moving sequentially through it, we're doing the production of NADH. Okay, so until we know the context of where that dehydrogenase enzyme is located, we can't say that we're producing NADH necessarily. Does that make sense?
What was your original question?
Which of those involve NADH or a redox mediator, I think is what I said. Because NADH is a bit of a lie here, because if we look at those four enzymes, see how well I memorized it. I believe 3 of them involve NADH and 1 involves FADH2. Okay.
And you can't, looking at dehydrogenase, you can't differentiate between.
So just because it says dehydrogenase, we can't differentiate between FADH2 and NADH. We need more context. Because dehydrogenase just says you remove hydride. Whoa, using what? Okay. Until we have more context of what that dehydrogenase is working on, we wouldn't actually know FAD or NAD. Okay. Pushing it further, even if we look at the name, isocitrate dehydrogenase, if we know nothing else, we just know the name of the enzyme, does that mean it's NAD versus FAD? No. The only way we would know that it's NAD in that case, forming NADH, is because we know that reaction, and when we clear everything away, we know that reaction involves NAD+. Okay, it's the coenzyme involved. Okay, so the dehydrogenase just means a redox reaction is happening. If we go back and take a look at all those that I highlighted, dehydrogenase, hydrogenase. The first one, there's our NAD. Second one, NAD, FAD. So let's go ahead and color that differently. And NAD. Okay. So 4 dehydrogenases, and in each of those 4 reactions, we used an electron mediator. Okay, or a redox mediator. Notice in the other ones, the name doesn't, at least as far as we've discussed it, does not directly mention anything about a redox mediator. And if we go back and look at those reactions, do they involve a redox mediator? No. Okay. So, kind of neat. We can use the enzyme name to help us identify things that are involved in it, or coenzymes that are part of it. Okay? Questions about that? Kayla, your face looks kind of concerned and passive.
I was thinking.
Okay, just checking. As far as the other enzymes, the other enzyme names, synthase shows up a couple times. We've got synthase and synthase. What are synthases?
Is that second one really, it's spelled different?
It's spelled different.
Synthetase.
It has a the
Oh.
in the middle.
Synthetase versus synthase. That's an interesting one. I actually don't know the difference there. Shoot, I can't underline that, which then removes my cheating through nomenclature.
Somebody was feeling fancy that day.
Yeah, that's quite likely that that's all that's happening. Because if we go through and look at those, it's citrate. Biochemistry is weird with nomenclature, so there might be a pattern here we can look at. If we look at the first one, citrate synthase, what did we make? What did we make in the citrate synthase? We made citrate. In the succinyl-CoA synthetase, what did we make? Succinate. What did we start with? Succinate-CoA. So maybe by throwing in that synthetase, that et there, they're saying, oh, we screwed up. That's actually the reverse reaction. That's thin. That's thin. Okay. I'm not familiar enough with how they come up with enzyme names to really play with that. Okay. But we've got some kind of pieces going on there. The aconitase and the fumarase. Aconitase is an interesting one because if we look at that reaction, what do we start with? Citrate, and what do we end with? Isocitrate. So why the heck is it called aconitase? Remember that one? Let's see, does the previous slide have it? Nope, not that one. Next slide? Yeah. So we are talking about, we said citrate, come on pen, is right here. Deisocitrate is right there. We're talking about that one reaction. Wait a second. That looks like 2 reactions. That's because it is 2 reactions. Right? We make an intermediate structure known as cis what? And that's my fault. Connotate. What was the name of that enzyme that did the citrate to isocitrate? Oh, a connotate. They're referencing the intermediate. Okay. Potentially because when that enzyme was discovered and they looked at that reaction, okay, so this reaction run in Krebs cycle goes to the enzyme aconitase, and quite likely what happened is when they pulled it out and studied it, they noticed citrate to cis aconitate, because aconitate is a stable intermediate. So they're like, oh, aconitase is being formed here, so let's call it an aconitase. And then someone was like, well, how do we go to isocitrate? And so they spent probably longer than anyone would care to admit or ever would admit, spent a really long time trying to find the enzyme that converts aconitate to isocitrate. And eventually someone played around with it enough and found out that it's the exact same enzyme. But at that point they've already named it, and so they can't change it.
You know the differentiation between the two? Enzyme synthetase.
Oh, you got it?
Yeah.
Sure.
By
What is it?
definition, synthetases must cleave or hydrolyze an ATP molecule to function, while synthases do not require.
Oh, that's fun. Which does not mean everybody uses that as your name, by the way.
Yeah.
So the synthetase, by definition, said we needed cleavage,
Hydrolyze an ATP molecule to
hydrolyze ATP. Anybody want to challenge that?
because it's GDP.
It's not ATP. In that reaction, it's looking at GTP. Okay. Not only that, you said the synthetase needed to cleave GTP, and what is it, or ATP, and what is it doing? And it's actually making it. So again, when it was discovered, they discovered the reverse reaction, so they named it according to that. Remember, your enzymes can go either direction. So we have to be very careful in the nomenclature associated with those enzyme names, because a lot of that comes down to how they were discovered. So the synthetase, what they're really talking about is not necessarily ATP. We're hydrolyzing a phosphate. Okay. Where that phosphate comes from is typically ATP, because ATP is our standard energy carrier. Okay. Why was it an energy carrier? Well, it had a high energy phosphate group at the end of it. We talked about that. Electron-electron repulsion, all that fun stuff. What happens if we made it GTP? Does it still have all that ion repulsion? Yeah. Okay. So we get the exact same things in GTP, causing it to be a high energy molecule that ATP is a high energy molecule. Why is GTP not the currency and ATP is? It evolved that way. Okay. Or if we really want to pull all of that out of it, we could say it was created that way. Might guess my opinions there. So, why use GTP instead of ATP? What was the point of the A or the G? That's not transferring the energy. The energy is all on the phosphate. So what is the point of the A and the G? I would bet the energy is pretty close, because it's all affiliated with that phosphate exchanging. Why A versus G? I've got 20 bucks. How are you going to get that 20 bucks from me? Try to be nice.
Is it the ATM thing you said? How, if you go to withdraw money from an ATM,
So that's referencing the energy,
right.
the energy value. The energy value for ATP and GTP are probably really, really close. Okay? There will be some subtle difference because they aren't the same structure, but they're, I would bet, very, very close in energy. So it's effectively still a $20 bill. We could go back to the key example. Okay? Your key unlocks a door. All you need is the part with the little jagged things to deal with the pin. If you jam just that part in the key socket, what happens? You are now locked out of your house. Okay? And it becomes very difficult for anybody to get in because you can't turn the key because all you've put in is all the pins. How do you turn the key? You need a handle. Okay? What is the A or the G acting as? The handle. That has different intermolecular forces that are going to interact with your enzyme differently, which is what's causing it to be used in one situation versus another. Okay? And for whatever reason, succinyl-CoA synthetase evolved to accept GTP as the handle, or the G as the handle as opposed to the A. Okay? And then fumarase, kind of like conatase, sort of. We have that ACE ending, so we know it's an enzyme. The first part of it is giving us some clue about when it's used. So fumarase, if we look at the first part, it's fumar. And if we look at, I said fumar, not bar. If we look back at the reaction for 7, we're reacting fumarate. Okay, so we have some kind of context of where that's happening. Kind of makes sense? Okay. I will say that when I took biochem, this was not a pleasant cycle. I killed many a tree with using scrapped paper and drawing out the citric acid cycle, structures, names, enzymes, all of the pieces, over and over and over and over until it was stuck in my head. I would highly encourage you to do the same thing. Okay? Because you will be held responsible. I'll give you as many clues as I can, but you're held responsible for recognizing what's happening at each of those stages. Yes? This was the conatase, and I did not do a very good job memorizing the enzyme names. This was a dehydrogenase. That says dehydrogenase. Just trust me. This was our synthetase. Right? I think so. This was another dehydrogenase. God, that looks awful. This was our fumarase. So those are backwards. I've missed one pretty badly in there. Oh, that has to be a dehydrogenase. This one's our fumarase. And then another dehydrogenase. That again says dehydrogenase. Okay. That is just the ending of those names. We need to clarify which dehydrogenase, so you'd have to go in and know the names of all those dehydrogenases. And does that really cover everything? No, because then we also need to remember that in the dehydrogenase, what else is going in? NAD, coming out is NADH. Okay. So there's a lot. Okay. If you want to go through and say what I had to go through and memorize, and you're probably doing something very similar. Let's see if I can find it. I think I've got it here. This looks horrible, okay? But a cleaned up version of this, that's what you're looking at, okay? I think I have, as much as I try to color code things, I have problems with colors, and this has way too much color on it for me. But that's effectively what you are trying to do with the citric acid cycle. You're doing the same thing when it comes down to glycolysis.
When you say a cleaned up version of that, are you talking about like just removing some of the clutter, like the structures?
Well, but responsible for structure. So what I would argue the cleaned up version is getting rid of all the color. Instead of doing space filling structures, just do the lines. So pretty much a crossbreed, it sounded kind of weird, crossbreed between this version and, come on, this version.
So like the structures on the other screen on this.
Overlaid on
the
this.
red.
Yeah. Because this gives you the enzyme names, okay? It also gives you that you have, when you have NAD plus and NADH coming in and out, okay? So I would argue that this is kind of the crux of all biochem kind of tests at what unfortunately is going to get called the superficial level. You'd get tested on everything happening within that. Okay. What we then layer on top of that is where are their important steps? Why are those things changing? How they're changing? All of that extra kind of fun stuff. Okay? Which I know seems super exciting, but that's what it is. Okay? Kind of makes sense? To get an idea of what kind of level of those questions are, this is why I would pull out the practice exam. Look at the practice exam, look at the questions that appear to be citric acid cycle E, and look at those. Okay. And within that, what I provide are the structures, the names, and then being asked for the enzyme. Or giving the enzyme and providing one of the structures, what's the missing one. Okay. And then giving you the structures and names as answer choices, okay, which sucks to provide, but I think it makes sense to provide it at that level because I don't think you should be forced to memorize it because it's easily found. Does that make sense? Okay. Yeah, so now let's look at it stepwise. Before we even get into the citric acid cycle, okay, so there's our citric acid cycle. To get in, we need the acetyl-CoA. Okay, that's how we start. So our pyruvate is coming in from glycolysis. Right there. To make our pyruvate, pyruvate can then, and this is why we have this weird split there, pyruvate can be used to make oxaloacetate. It can also be used to make acetyl-CoA, and acetyl-CoA is our lead-in to our citrate. Okay? Those are two different enzymes, two different reactions, and depending on the availability of those biomolecules, we may end up using pyruvate to make oxaloacetate, we may end up using pyruvate to make acetyl-CoA. It's going to come down to what pieces are readily available at that time in that situation for the sup. Okay, so to get this is totally not the Onramp, well, it's kind of the Onramp. It's our first step in our citric acid cycle. Okay, we're taking our oxaloacetate, we're reacting it with acetyl-CoA to make citrate. Okay, this reaction is exothermic, and because it is exothermic, that is why we don't need to use ATP. Okay? So it is a reaction that will happen on its own. Why might it be exothermic? Kind of a harder application question. We've covered it. Subtle hint. Becoming a less of a subtle hint. That bond is insanely high energy. The sulfur is a great leaving group. That structure can get polarized and we can break that bond. Okay? Breaking of that bond releases tons of energy. Okay? How the bond breaks becomes the next part that we have to address. Okay? But it's that bond breaking that's really powering this whole process. So when we went back and looked at, if you go back and look at coenzyme A, the reason we have coenzyme A is because we make that high energy bond. So we don't need ATP because that bond is the one that supplies most of the energy to drive this reaction. Okay? Which is kind of a weird statement to make. Really what we're saying is that that coenzyme A changes the energy dynamics of this piece enough that it becomes reactive enough to easily react with the oxaloacetate. Okay? So it just changes the energetics of what can happen within there. Okay? Kind of makes sense? Okay. Because we talked about worrying about inhibitions, we take a look at our text. NADH, ATP, and succinyl-CoA will all inhibit this reaction. Why would I expect NADH and ATP to kill it?
You don't want to start in the cycle.
Okay. What we're looking at is that, whoa, why did my screen freak out? That was weird. That's never happened. Hopefully it doesn't die. We're looking at this first step. Okay. So we're talking about using acetyl-CoA, which was an awesome synthetic reagent because it's high energy and we can dump acetyl groups pretty much wherever we want now. Okay. And oxaloacetate. If those are synthetically useful, or officially even pyruvate, if those are synthetically useful molecules, right, I don't want to dump them into the citric acid cycle if I have plenty of energy, because the point of the citric acid cycle is to get energy. Right? So I want to prevent acetyl-CoA from disappearing into the cycle if I can. Okay? So that's why NADH shows up, because NADH is a big leader to producing energy. Well, as it produces energy, the energy it's producing is in the form of ATP. So both NADH and ATP are going to come in and potentially inhibit this enzyme. If I have tons of NADH, that NADH can be used to make ATP. If I have tons of ATP, well, I don't need energy, so shut this thing down. Okay? That means those pieces can then go out into their respective environment and be used for something else. Want to make sense? How about succinyl-CoA? Well, that's because we started with succinyl-CoA, right? No. Okay, so we ended with it.
Is that what that coax is then? At the end?
No. That is coenzyme A. So I see what you're saying. The S could be succinyl. No. The S is not succinyl. The S is sulfur.
So that succinyl-CoA is not.
It's not involved at all whatsoever in the reaction. But neither was NADH or ATP. If we go back and look at our reaction, succinyl-CoA is further through the chain. Okay. If we go through, citrate turns into D-isocitrate. Okay. Does that produce any energy? No. Isocitrate to alpha-ketoglutarate. Does that produce any energy? Okay. Hard again to look at the structure, so we could take a look. Isocitrate to alpha-ketoglutarate? Yup, that gets some NADH. What about succinyl-CoA? In the very next step, succinyl-CoA produces NADH. And once I form succinyl-CoA, I can get GTP, which is another direct energy currency. So if I have tons of energy, okay, I in theory have a ton of GTP. Well, then what do I want to not make more of? GTP. So I better not have succinyl-CoA floating around, because succinyl-CoA is going to do what? Make GTP. So succinyl-CoA, even though it's way later in the reaction, can also be used to shut that reaction down. Okay? So we get lots of different ways that those molecules can come back and inhibit and prevent our reaction from happening. Okay? Why else might we want succinyl-CoA to shut that down? What was that piece? Coenzyme A, which happened to generate very high energy sulfur or thioester bonds. The same thioester bond that we have in our acetyl-CoA. Okay? So we're trying to kind of limit that reaction from happening, okay, with whatever pieces we've got. Kind of make sense? Okay, so we've got our inhibition. Now the question on everybody's mind is, when is Mike going to bring in OChem? Here you go. how does acetyl-CoA react with oxaloacetate? So what changes between oxaloacetate and citrate? And number one, easiest question, why would I pick oxaloacetate and citrate as the difference as opposed to, say, acetyl-CoA and citrate? Why am I asking to compare oxaloacetate and citrate and not acetyl-CoA and citrate? It's an easy answer. There's a larger structure of oxaloacetate showing up in citrate than there is of acetyl-CoA showing up in citrate. Right? So it's easier to compare the difference of the biggest pieces and saying, okay, now how did those big pieces change? Well, the little thing's going to have to do something. Okay? Kind of makes sense? Okay? So oxaloacetate to citrate. What might we need to find? Okay? So you're saying you lost a bond to this top oxygen. Okay, so you're... Okay, so I would argue before you start trying to go through and say what bonds are breaking and making, you should find oxaloacetate's main structure in citrate. Okay, so we've got those 3 carbons. So there's those 3 carbons. And then it turns. And then it turns. Right. So that's our structure, oxaloacetate to citrate. There it is. Which means where's the chemistry happening? At that carbonyl carbon, which we'll go ahead and call purple for this moment. Which then means the, let's call it a green oxygen, is likely which oxygen on the far right? The OH. So what happened between the purple carbon and our green oxygen in the course of the reaction? It lost a bond. Okay. Where would I expect the electrons to go? So if we look at the red bond on the reactant, those electrons should go with the carbon or with the oxygen? The oxygen, because oxygen is more electronegative, which means the oxygen is carrying what charge? A partial negative, and the carbon is carrying a partial positive. Okay. How does that help us out? Well, that partial negative would likely pick up a positive from our solution. What is it picking up? It's picking it up from water. What atom? It's picking up, I want a little bit more than hydrogen, a positive hydrogen. So we're getting that from our water. So our enzyme is somehow helping water and cleaving water to dump a hydrogen on there. Okay. What's happening at the carbon? Well, it is positively charged, so what does it need to react with? A negative. A negative what? Picks up a carbon. So a negative what? A negative carbon. The carbon in our citrate, the blue carbon there, must have come in as a negative. Because when we look at oxaloacetate, that purple carbon was positive. So citrates, or our blue carbon, has to come in as a negative. That blue carbon is coming from where? From acetyl-CoA. Where's the negative carbon in acetyl-CoA? CH3 is negative? No. So we could say the carbon attached to it. So you're saying that is my blue carbon. That has an electronegative oxygen pulling electrons away and an electronegative sulfur pulling electrons away. That carbon is most decidedly positive. So that can't possibly be the blue carbon. It has to be the CH3. What's the problem with the CH3? It is definitely neutral. Okay? Not negative. Well, maybe we break a bond.
Wait a minute.
What?
Would that negative oxygen take a hydrogen from that carbon instead of water?
The specifics of where it is we don't need to stress about.
But
But let's focus on what you're suggesting there. You're saying that we remove a hydrogen, and then that would then make that carbon negative. But we're then breaking a carbon-hydrogen bond. Are we allowed to break carbon-hydrogen bonds? Why not? They're nonpolar, they're stupidly strong bonds. That is not an easy bond to break, unless
The alpha carbon.
it happens to be at the alpha carbon to a carbonyl, because we now have resonance generating. You can do better than that. Son of a. It's an enolate. Right? This is why finding the enolates is insanely important, and it needs to be probably a more important aspect in organic, because the enolate chemistry is what's actually driving this reaction. Okay? That hydrogen on the alpha carbon, that CH3, is actually acidic due to resonance within the structure. We have an enolate. That enolate is what can come in and attack our carbonyl carbon, breaking that open. We're now darn close to the citrate. Except what would we actually have? Okay, we still have the C bonded to O, but then we would have S-CoA. What else was floating around? What did we remove from the water? A hydrogen. So what's left from the water? What charge would that oxygen be? Negative. A negatively charged atom is a pretty good nucleophile. Well, Mike, that's just becoming more of a mess. Except what is special about the sulfur? It's a phenomenal leaving group. Which then means we would end up with, surprise of surprises, citrate. This is neat. I like that. Right? It's looking at all of those pieces, and it is fascinating because you look at the overall chemistry to start with, and you'd be like, everything's positive. There's no way in heck you can make a carbon-carbon bond if everything's positive. Oh, we've got an enolate. That still doesn't fix it. Oh, but sulfur's a great leaving group. I've got a leftover piece from water that can come in and kick out the sulfur. Okay? That is pretty freaking amazing. If you were asked to draw out a full-on mechanism for this, it gets stupidly long. Right? What's happening within it? All of this is happening in fractions of seconds because the enzyme brings all of these pieces near each other and says, okay, go. Spits out a product. It's nuts. It's absolutely crazy nuts. Right? And for those of you who think I'm excited and think this is all neat, it is neat. I don't want to study it. Okay? It is really, 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 really cool, okay, that it can go through and do this. Right? So what we need to be aware of when we're looking at this is then how can we kind of test all of these aspects. So number one, we need to know the structures. We need to know the enzyme name. We need to know what's happening within that. Okay? The next part is recognizing where the pieces go. Okay? That's what we highlighted when we looked at our structure and said the oxaloacetate structure is showing up over here. Which then means the acetyl-CoA, 2 carbons, are right here. So it's recognizing where those pieces go in our sequence or in our reaction. Okay? The next part, because it's me, sorry, is we also have to recognize the importance of how the reaction is happening. That means the oxaloacetate is acting as an electrophile in the course of the reaction because it is accepting electrons. That was our positive carbon. The acetyl-CoA acted as the nucleophile, and it was able to act as a nucleophile due to the resonance found within the keto-enol tautomerization. So the next time someone tells you that biochemistry doesn't involve organic chemistry, you can slap them. And you can point to this reaction and be like, you don't know what you're talking about, dude. Or dudette. Okay? Kind of makes sense? Right. Getting into the nitty-gritty on the OChem, I am fully, again, aware that we may not all be up there. That is one of the higher level questions. The bulk of the exam is not at that level. The bulk of the exam is at that superficial level. These are the pieces. This is what it becomes. Okay? So you should absolutely be able to pass the class by saying these are the pieces, this is what it becomes. Okay? If you want to advance further up, at least on this version of the exam, you need to watch out for labeling electrophiles, nucleophiles, and recognizing where those pieces are. Does that make sense? Step 1. So, next one. Right, the enzyme aconitase. Right? So let's take a look at the difference between citrate and D-isocitrate. I just moved an alcohol group. So they are technically isomers. Okay? So let's look kind of briefly at what happens. We start with citrate and we go to cis-aconitase. What kind of reaction did we do there? We did an elimination. How do we know we did an elimination? Well, remember I said there were the reactions that you needed to know. What was the product of that first step? We made a pi bond. Okay? Which in this case is an alkene. I would back away from the functional group name and stick with the actual bond, pi bond. Okay? Which means we did an elimination. Okay? What did we eliminate? Okay? Almost everybody sees the hydroxide. Okay? But we really eliminated from the molecule water because we had to get rid of a hydrogen from another position. Now we've got our full elimination. We broke 2 sigma bonds. We eliminated water from the structure. We now have the cis-aconitase. Aconitate. God. That's aconitate. The enzyme is the ACE. Okay. Then what do? We do an addition reaction. So in our elimination, we lost H2O. What did we add? Really? Yeah, really. We lost water, we added water. So the enzyme, interestingly enough, does 2 things. Well, all enzymes do 2 things. They do the forward and they do the reverse. But this is doing the forward, getting rid of water, and the reverse, adding water back on in a different spot. Okay? So this enzyme theoretically has the ability to do 4 different things. Remove water to form the aconitate, add water back to take us back to the citrate, right? Add water to the aconitate to make us D-isocitrate, right? Or remove the water to take us to the aconitate. Okay, so that's kind of neat. We can do a whole bunch of different things. So why?
Is it to get the hydroxyl group out of the kind of the steric strain of those functional groups and kind of move it around?
So that could shift the hydroxyl group out. So I like that idea. That gets the OH kind of out free and clear. Okay, but why would I want the OH out free and clear? What happens in the next step? I convert the alcohol into a ketone. Couldn't I have converted the alcohol into a ketone at citrate? Why not? It's not sterics. If we tried to make a pi bond there, what happened? We'd exceed the octet on our central carbon. Okay. Well, then we could kick this out. Is that a good leaving group? No. CO2 is great as a leaving or as a group that can leave the structure, but by doing arrow pushing the other direction, which would then be all sorts of problems with electron density. So we just have too much electrons smacked in the citrate center, so we end up pulling that out just a little bit to get us to the isocitrate structure. That opens up chemistry on the alcohol. I can now do oxidation reactions at the alcohol. I couldn't do any oxidation reactions on the citrate because the alcohol is the only thing I can oxidize, and it's a tertiary alcohol. All of the other carbons are as oxidized as they can possibly be easily. Okay, so by converting into the isocitrate, I now have some oxidation chemistry available. Make sense? Son of a, stop saying that. What is special about isocitrate?
It's D.
It's D. What is the D referencing? Chirality. Why did we not call citrate D? There's no chiral atom. To have a chiral atom, we have to have 4 distinct groups. Okay? Where the alcohol is attached does have 4 things attached, but what is the difference between that carbon and that carbon? There isn't one. Okay? When I shift the location of the alcohol, now what happens? Well, those carbons are different. I also have a hydrogen out there, so there's 4 different locations. That is a chiral atom. What else? That is a chiral atom. Which means in the process of doing this reaction, I have 4 possible products that can come out of this. Right? Because we have 2 chiral atoms. Okay? That makes a big mess of things. What does the enzyme do? It only makes one of those 4. Which one? D-isocitrate. Okay? So we have to reference that accordingly. That's why we're bringing the capital D in there. Okay? Is that capital D the same thing as R and S? No. What is the D in reference to?
Isn't it visible?
Fisher.
It's in reference to the Fisher projection. Okay? We're looking at the second to last chiral atom. The highest priority atom on the second to last chiral atom is located on the right-hand side of the Fisher projection. Yay! Okay? I just wanted to remind you of that. Okay? I'm not testing on that. The part I'm testing on is that that is chiral, and we need to know that that's D-isocitrate, and that's why the D is popping up. I don't expect you to do all the Fisher projection nonsense again. Okay? So, we now have D-isocitrate. We're going to convert that to alpha-ketoglutarate. And I had issues with fonts, so it looks like A. But just pretend that says alpha. What changed?
We lost some stuff.
Yes, we lost some stuff.
And there's a read.
Okay. This group has disappeared. Ooh. Somebody may have just given you a hint about that group disappearing on the previous slide. What else happened?
There's a redox reaction.
There's a redox reaction at the chiral center. Describe that a little bit more for me.
The alcohol.
The alcohol is converted into a ketone by doing what?
Gain.
Elimination is where I was going. All of those answers were correct. Oxidation. What are we losing? But H plus isn't a big deal. That's not an oxidation.
We gain electrons to oxygen.
What else do we lose? Nope. Well, yes, but no. I don't care about the CO2 right now. I care about everything in purple. What else did I lose? Yes, that hydrogen came off. What? Who did we lose?
Hydrogen, carbon.
We lost another hydrogen?
That's a carbon, yeah.
Let's call that our blue hydrogen. We lost a hydrogen? Anyone wanna... We lost a hydride. Okay? Remember... Oh, don't lose connection. No. You Maybe. You don't suck, right? Yeah, good. This is all the fun stuff. And the stuff that's not in the textbook, so we need this recorded. Okay? We're losing a positive hydrogen out on the end. How do we know that's a positive hydrogen? Well, where would the electrons go between oxygen and carbon? To the oxygen, which means those electrons can swing down to form the pi bond. Well, between carbon and hydrogen, where would the electrons go? We might predict the carbon, okay, because it's slightly more electronegative, but then that's electron-electron repulsion. So the electrons end up getting forced out onto the hydrogen, which means we have H minus. And as soon as you say H minus, you should be saying NAD, officially NAD plus. So this reaction will need NAD. Okay. If we go through and look at the stuff going here on the far side, it is another allosteric enzyme. Okay. Remember, NADH is something we can produce energy from. It is going to be inhibited by NADH because if I have a bunch of NADH floating around, I don't want to make more. It will also get inhibited by ATP because that's energy. The whole point of this is to produce energy. So if I have a ton of energy, don't do it. Okay? Let's address the pieces of this because there's officially two things happening here. Okay? We've got the purple part and the red part. Okay? And this is a neat reaction too. Okay, so the purple part, we already kind of have some arrows on, okay, dealing with the NADH. But that really has nothing to do with the CO2. So what's going on with that? Well, CO2. How do we turn that red part into CO2? Put the electrons there, and that would then put the electrons there, which would mean we have a negative carbon. Can I have a negative carbon? No. Which means CO2 can't leave. How can I get a negative carbon? I have to form an enolate. Right? Can I form an enolate and diisocitrate as it's shown up there? I would have a negative carbon next to an alcohol, so no. So let's, because the structure's already partially drawn for us, let's go through and do the oxidation reaction first. By doing the oxidation reaction first, I now have the carbonyl. So not alpha-ketoglutarate anymore. I now have this guy. Now what happens if those electrons come down? I put a negative carbon again. So, well, Mike, that's the negative carbon. Except now what do I have? I now have a negative carbon next to a carbonyl, and I have resonance in the form of the enolate. So there's two big things happening here. We're kicking out CO2, officially respiration, and we're doing the oxidation reaction. Those are the two primary reactions happening. The order is critical here. We cannot kick out the CO2 first because we don't form stable intermediates. So what happens? We do the oxidation first, then we can kick out the CO2. Okay? So yet another kind of amazing enzyme because it's doing two massively different things, but it's doing them kind of simultaneously. Make sense? Should we write that down again? So let's look, we'll call it purple and red up at the top. Okay. We have an oxidation happening in purple, and then in red we have what is known as a decarboxylation because we removed a carboxyl group. Okay? So we have to do the oxidation first, then we can follow that up with the decarboxylation. Okay? Yes, I can ask a test question on that. Okay? The mechanistic details on how the enzyme is doing that, what amino acids, all that fun stuff, don't stress about. Okay? But recognizing what things have changed is an important thing to do. So we're losing CO2, we're doing an oxidation. Because we're doing an oxidation, we need NAD. Okay? The decarboxylation is kind of a secondary feature of that.
Does it
I don't care. Let's put it that way.
This one is.
So yeah, what I would probably do is which of the following molecules is likely involved in this enzyme reaction? NAD plus, ATP, NADH. I probably wouldn't say coenzyme A. GTP, because that's in there. So that gets me 4 answers. Typically there's 5 peanuts. Okay, so peanuts clearly is wrong. Okay, ATP and GTP. There's no phosphate transfers, so those don't make any sense. Now it's NADH or NAD+. Okay. We need NAD+ because we need something to accept the hydride, the H- coming out. Okay, makes sense? So you could look at it by looking at the structures, or you would know that answer because you memorized that whole cycle. Make sense? Okay. And see, there's our NAD+ and our enolate formation. Step 4. Okay. Actually, let's go back to that. What enzyme do you think we'd use for that? Uses NAD+. Dehydrogenase. Next one. What changed?
Changing the functional group.
All right, this one actually gets pretty nasty, I think, personally.
Get rid of CO2,
That CO2 has disappeared from the structure, and we brought in coenzyme A. Okay, which is nice because the sulfur, that's an O, is nucleophilic and could potentially attack. But wait a second, wouldn't those electrons come back down in, then pushing those electrons out? Doesn't that mean we have way too many electrons there? Yep. So if we have an excess of electrons, what do we need? We need something to accept those electrons, like NAD, okay, which this one I think is actually FAD. I could be wrong on that. Okay. But we need an electron mediator to help with this. This one's less obvious, in my opinion, obvious is a loose term, than the last one, because the last one we kicked out H-. Here we just say, holy crap, there's too many electrons there. Okay. So what's happening within this? Well, again, it's going to be a dehydrogenase enzyme, which is then going to require NAD+. Okay. This one is a big nasty one. You're actually dealing with FAD, which will then eventually turn into NAD+. Okay. Because of how many different steps that are happening within this. Okay. So this little drawing is trying to show how technical. Oh man, that's a horrible drawing. How did I not see that? Sorry. Is showing actually the reverse reaction, NAD dumping hydrogen to FAD. Okay, but we're really looking at kind of the reverse here. Okay, so this uses our thiamine, the lipoic acid, the FAD, the NAD+. Okay, which we saw already in which enzyme? That was that big nasty wheel of stuff. Not in the citric acid cycle. Showed up actually in 2 places. The pyruvate dehydrogenase to make our acetyl-CoA, and the other one, this is why beer has lots of B vitamins. Our B vitamins are thiamine. Why would beer have lots of thiamine in it? The anaerobic respiration with alcohol, or technically, it's not pyruvate dehydrogenase. It's is it that? Maybe they call it the acetyl. I don't remember the name of it. Okay, but it's used twice. The anaerobic respiration enzyme, okay, and our pyruvate dehydrogenase. This is a similar process using a different enzyme because they're different substrates, but we're still going through using thiamine as our Schiff base, to help polarize our structure to initiate all of this. That will then dump it off to lipoic acid, which dumps it off to FAD, which then dumps off to NAD+. Okay? So this is, again, a massive complex dealing with all those individual pieces, all sorts of fun moving parts. Okay? The big part that I want you to know here is recognize the changes. Okay? We need an electron mediator for this reaction. Okay? Not only do we need an electron mediator, what else is produced? We also lose the CO2. Okay? So it's a redox reaction, and we've lost CO2. It's interesting. This was in our oxidative our oxidative process consuming oxygen. Okay, well, we haven't consumed oxygen yet. The oxygen reacts with all of our effectively electron carriers with our NADH. Okay, so what else is happening in this process? Well, when we consume oxygen, we're reacting with NADH. How did we get the NADH? We expelled CO2. This is where our CO2 is coming from when we expel stuff from our body or expel air. Okay. Which takes us now to our succinyl-CoA, and we convert that across into succinate. So, big changes. What did we do? Okay, this piece falls off, but the only way that piece could fall off is releasing energy. But then if that's coming off, it's coming off with the electrons, which means that carbon is really, really positive. That ain't stable, guys. Fix it. What could come in? Well, look at your product. Where is that carbon in the product? Right. That COO minus, particularly irritating. Okay, it's our carbonyl, which we already had. It's an oxygen. An oxygen is coming in, okay, which we could get from, say, like water. Okay. Interestingly enough, there's no water. What's coming in?
Could be an oxygen phosphate.
Our inorganic phosphate can dump electrons in, kicking out our group. But then what we would end up with is not the structure shown for succinate. We would have that stupid phosphate hanging off of that, and that's not what succinate is. So what happens?
It gives an oxygen, but then it goes and attaches to GDP.
GDP has a free oxygen hanging off on the end. So to break off the succinyl-CoA, it releases enough energy that we can dump a phosphate, an inorganic phosphate, onto the structure. Once the inorganic phosphate is attached, I can then take that high-energy phosphate bond and dump it onto an energy carrier. That energy carrier, in this case, happens to be GDP. Okay? Not ADP because, again, the handle. So we end up producing energy, GTP, in this process. We take advantage of the high energy thiol bond. I still got time? Come on. I'm usually late.
They're in your clip.
That high energy thioester bond is replaced with the slightly lower energy phosphate bond, which we can then dump onto our energy carrier. Cool. Okay. And what kind of reaction? I know what I'm thinking of in my head, but I don't think I asked the question right. Say that, because you're coming up with the answer, yeah. It is a phosphorylation, but I don't quite accept that as the fully complete answer. There are two types of phosphorylations. Oxidative phosphorylation. Is this oxidative? No, we didn't use any redox mediator, so this is not considered oxidative phosphorylation. It would be substrate level. Okay. So succinyl-CoA synthetase, because we're producing, that was back to what the synth-et was referencing. It's our production of energy. Okay. And we're now at, if we clean that all up, can I clean that all up? Okay. Our succinate. Cool. We've reformed our coenzyme A that can go back through our cycle and react with something else. Our GTP now has energy that could potentially go out and donate that energy somewhere else. And we're now to succinate. Okay? Yeah. Okay. What? Oh, I know why that's here. So that says step 5, right? We're talking about it just kind of does all that fun stuff. What was happening in this thing? This looks kind of familiar, right? This was our pyruvate conversion, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. What happened? That was in our glycolysis. What happened in that reaction? We added a phosphate. Okay. Why would I bring up this step in comparison to this step? We're still adding the phosphate, but this one required an electron mediator.
So wait, before we read further, this is not part of?
This is not part of
This is
Krebs cycle. This is glycolysis. This is pyruvate conversion. We're just showing it as saying, what are the similarities? Because we're doing a lot of similar reactions.
Is it the two differences between phosphorylation? One's substrate, one's...
They're still both substrate level phosphorylation.
Wait, this one's not oxidative?
This one is oxidative because it has NAD+. The other one was not oxidative. Okay, why did this one need to be oxidation, but this one did not?
That one has a good leaving group.
It has to do with the leaving group. Okay. In step 5 of our Krebs cycle, oops, sorry, I shouldn't have done that. Those of you watching at home, I apologize. Don't get sick. That sulfur was a great leaving group, okay, which is what facilitated all of that. In the pyruvate dehydrogenation, where's our chemistry happening? Carbon number 1, did we have a good leaving group? No. Okay. So in this case, we needed cysteine, and cysteine had sulfur. So the enzyme is supplying the sulfur that coenzyme A is supplying back here. Okay. So it's still pretty much doing the same chemistry, but this one has an intermediate that we can manipulate. In pyruvate conversion, the enzyme had to make that intermediate using its structure. Okay. So neat parallels are happening between them. That's really all I was trying to do. Whoa. Okay, I don't know why I did that. As far as our conversions, the formation of our ATP will always be endergonic. Okay. Can't quite say it that way. ADP to ATP is endergonic. By bringing in a high energy phosphate compound, we can make the reaction exothermic. Okay, so depending on which part of the reaction and how we're labeling it, we'll reference them as exo or endo. Okay. Jumping into 6 and 7, okay, we now have our succinate, which we convert to fumarate. What's the difference? We did an elimination reaction. What did we eliminate? Okay. We eliminated hydrogen. Okay. I heard reference to water. What oxygen was removed when we did succinate to fumarate? There's no oxygens that are removed. It's just hydrogen. So just look to the succinate to fumarate, not the malate. Okay. If I remove hydrogen, I could probably remove an H+, which would make this carbon negative. Is that a stable negative? Technically, through an enolate, we can't do enolates well with carboxylic acid, so the enzyme is going to have to facilitate and allow that to happen, just so you know. Okay, but let's just say we form that negative. What does that mean the other carbon needs to be? Positive. How does that carbon get positive? What are we kicking out? Hydride. Ooh, hydride means NAD+. Okay? So we need another redox mediator to facilitate that reaction. We can convert succinate to fumarate. Then what do? Then we do an addition reaction. So this was our elimination. Elimination of H2. Therefore, if you haven't seen the 3 dots, that means therefore NAD+. And the next one, we're doing an addition reaction. What are we adding? We're adding H and OH. We're technically adding water across the pi bond. Okay. Which is starting to get us really familiar looking. What does malate look like? Because you got the structures all memorized by now, right? Remember, this is a circle, and this is looking awfully close based on my arrows on getting back. Looks like oxaloacetate. What's the difference between malate and oxaloacetate? Dun, 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 dun. It's a ketone versus an alcohol. So, and our next step, our malate is converted into oxaloacetate, okay, by making the ketone. To get the ketone, what do I have to do? I have to take electrons over here to make the pi bond, but then what would I be kicking out? Hydride. As soon as I see hydride, NAD+. And all the way back to oxaloacetate. Now what can I do? Start all over again with acetyl-CoA, which reacts to make citrate. And we can now start our whole cycle. Okay. This step is not inhibited by NADH, FADH2, or ATP, which is interesting because what does it produce? NADH. So why might it not be inhibited? That's a really interesting question. One we'll pick up with Tuesday.